Coastal zones are one of the ocean's most productive environments, and they support major subsistence, industrial, and recreational fisheries. These regions face growing and compounding threats from many sources, including overfishing, climate change, and pollution. Now, we study food web structure because food web structure mediates the impacts of these threats on coastal ecosystems through its effect on essential properties such as productivity, connectivity, and energy transfer efficiency. Understanding the structure of coastal food webs is an essential element of ecosystem-based management, predicting responses to overfishing and climate change, and tracing the bioaccumulation of contaminants. An accurate account of a food web structure is also necessary for ecosystem models to parameterize and establish model boundaries. But one of our main findings from this research was that increases in primary production led to shorter food chains in eutrophic coastal ecosystems. This is interesting because the classical hypothesis in ecology is that higher productivity should lead to longer food chains because there is more energy available to support higher trophic levels. Now, we have long known that an opposite effect is seen in marine environments, where in low nutrient, low productivity areas, such as the high seas, the highly size-structured nature of pelagic ecosystems meant that lower productivity produced longer food chains through the need for more trophic steps to process the small-sized phytoplankton associated with these regions. In productive regions, for example, like eutrophic coastal regions, the focus of the study, where the size structure of phytoplankton is inherently larger, there was a mix of hypotheses as to how greater primary production may impact food chain length, whether it was lengthening it, shortening it, or having no effect. In this paper, we demonstrate how increases in primary production can lead to increases in secondary zooplankton production and cause increases in omnivory, which is foraging at more than one trophic level, in that the higher trophic level organisms, the predatory fish higher in the food web, will start foraging more on these lower trophic level zooplankton prey, truncating the food web and shrinking food chains in highly productive coastal regions. This study relied on stable isotopes to make these conclusions. We use stable isotopes to estimate trophic levels and organisms' position in the food web, whether it's trophic level 1, autotrophic phytoplankton, or 2, a zooplankton that eats phytoplankton, or trophic level 3, a fish that eats zooplankton, and so on. Stable isotopes are chemical tracers. They're innate properties that can be measured in all components of a food web, from phytoplankton to large predators. Because their values are altered predictably as they move through the food web, as one organism consumes another, by measuring them, we can get an estimate of an individual's place in the food web, its trophic level that I mentioned earlier. Stable isotopes are also measured in tissue or material that is built up over time, meaning the isotopes provide a time-integrated signal of foraging. If we sample an entire food web in a discrete window of time, we can get a time-integrated snapshot of the food web's trophic connections and the position of every individual in that system. By doing this for multiple food webs here along the BC coast that shared similar species but varied in their baseline levels of primary production, we were able to identify differences in trophic level within the same species across regions, which showed us that there was greater levels of omnivory happening in regions with greater primary production. The length and structure of marine food webs has implications for ecosystem connectivity, efficiency, and fisheries productivity. Developing a clearer understanding of what controls food web dynamics in eutrophic ecosystems allows for better modeling and predicting. Again, these are some of the ocean's most productive regions and are home to some of our largest and most significant fisheries resources. As these regions change, whether it's from climate change impacting levels of primary production or overfishing changing prey fields, it will be crucial to have a clearer understanding of how trophic relationships will change, as these can impact energy efficiency, diet quality, nutrient uptake, and contaminant uptake. This research helps bring this picture into clearer and sharper focus, giving us the ability to update our ecosystem models as conditions change and anticipate changes to diet and predation.